Hello friends, good afternoon. Welcome to CEC live lectures. Dear friends, in this session we will be continuing with our ongoing lecture series on photosynthesis. In the series, today's the topic for discussion is mechanism for photosynthesis. The topic is divided into two parts, and today we will be dealing with the part 1, in which we will be discussing on nature and fate of light in photosynthesis. To take this topic, we have with us our subject expert, Dr. Eklavya Chauhan. Dr. Chauhan is Associate Professor, Department of Botany, Deshbandhu College, University of Delhi. So without wasting much time, I would like to welcome Sir in our studios and request him to start the lecture. Welcome, sir.
Thank you, Asmita. Dear viewers, we have been dealing with this topic for the past 2 terms, and if you remember, in our first lecture we had talked about the entire gambit of the history of photosynthesis and how it took a long 2.5 centuries to reach a tangible equation or a correct equation for photosynthesis, which, although could be further simplified to its empirical form, yet we signify 6 molecules of water to say that it is a biological equation, because the sources of oxygen which is being released in photosynthesis and the sources for carbon in sugars, they are quite different. Today we have to dwell upon the idea as to how light triggers the most vital phenomenon of photosynthesis. But before that, we have to have a recap of what gives what and which gives rise to what. So the correct equation, as you know, is based on the earlier ideas of C.B. von Neil, which suggests that the carbohydrates are synthesized from carbon dioxide and water. But mind you, this water is not the water which is being used in photosynthesis as a raw material, only some of it, and that is the oxygen part of water is being entirely utilized for the formation of molecular oxygen. So the one which is liberated comes from water, and the oxygen of carbon dioxide is responsible for giving the oxygen of the sugar component as well as the 6 molecules of water which are being liberated. So as to say, the oxygen of carbon dioxide in fact is being ramified into 2 products, namely the hexose sugars as well as the water which is being synthesized. But the vital question which got solved and finally the whole controversy was laid to rest was that the source of molecular oxygen released in photosynthesis was actually water and not carbon dioxide. Just reminding ourselves of the early experiments of Robin Hill in 1937, while he studied the redox reactions of photosynthesis. Most of his experiments were performed in vitro. So he isolated live intact chloroplasts, and he gave them light, and he assured that the carbon dioxide in the medium was not there, and then he added an oxidized electron acceptor. In those days, the electron acceptors he used were ferricyanide and benzoquinone, and of course he provided them with water. And what he saw as the product was that these acceptors, they immediately got reduced. Now this reduction was perhaps gauged by a change in their color, say a typical redox reaction, and in the process oxygen was being produced. Now this was a very important phenomenon to suggest that even the isolated plastids could bring about the production of oxygen. However, no sugar was produced during this process. Further, when the chloroplasts, the isolated chloroplasts, were kept in dark and all these conditions were also provided and duplicated as they were. As far as the illuminated side was concerned, the product was very different. It showed that in this case there was no water, there was no oxygen which was being released. Now this further enthused Hill, and his work was then further elaborated by Arnon, who was also a Nobel laureate later. And in the presence of light, when carbon dioxide is absent, and the question is, if ADP or NADP is provided and water is provided, the products are going to be ATP and NADPH. So that means the story got a bit further, that oxygen is produced and so are these two products. So perhaps what Hill performed in vitro, Arnon duplicated, or he simulated the type of acceptors which might be existing in nature in the plastids. However, no sugars were produced during this time also. And finally, when the chloroplasts, they were kept in dark after isolation, they were given ATP and NADPH and H. You remember, these were produced when the chloroplasts were kept in light or they were illuminated, and then in addition carbon dioxide was also added. The products now were ADP and inorganic phosphate plus NADP. So therefore some sort of a oxidation reduction process was going on, and in the process sugars were produced. So this led to very important conclusions, and that is that Arnon had actually separated one reaction which was light dependent, which was producing ATP, NADPH and oxygen, and there was another reaction which was separated, which was a light independent reaction, which was to be called as dark reaction later, whose sole aim was to produce sugars. However, sugars would not have been produced if ATP and NADPH were not provided to it. So this was the beginning of the appreciation of two components of photosynthesis, namely a light reaction which was a photochemical reaction that is dependent on light, and the other one, the dark reaction, which was as we saw in the previous lecture, which was dependent on temperature, having a Q10 or a temperature coefficient of 2 to even 3. So to summarize the whole work, the isolated live plastids from the plant of Stellaria media, when illuminated in the presence of electron acceptors like ferricyanide, they evolved oxygen. So these were artificial hydrogen acceptors. In nature, the acceptors are different. They are in the form of NADP. So that means there are actually two separate processes which are involved in the photochemical evolution of oxygen. Don't you think this gives us an idea of an inverter being charged with electricity, and then when the light goes off, then the inverter itself provides electricity and itself gets discharged only to get recharged later. So this is a very crude analogy but a very effective one. So in nature it is NADP which is the acceptor of electrons and functions as a reducing agent. So that means once NADP is reduced to NADPH, that is the reduced coenzyme 2, then only it would be effective in the Calvin cycle reactions or the reactions of the dark reaction, which are going to produce ultimately sugars. So in a nutshell, there are two stages of photosynthesis: the light reaction or the photo stage, which is going to be entirely dependent on light, and this light is going to trigger energy fixing reactions, and they are going to convert light energy into ATP and NADPH to drive the next stage. These are very complicated processes, and then followed by the dark reaction. Now this reaction is what we call in terms of Calvin cycle, the Benson-Calvin cycle, or the C3 cycle, where a whole lot of carbon fixing reactions which are going to utilize ATP and NADPH, which were coming from the light reaction, to convert these inorganic molecules into sugars. In other words, the dark pickup of carbon dioxide can also be likened to something like an organic fuel, which stores potential energy in the bonds. This is how the sugars are made. So what would be the rationale for light reaction? We have been saying repeatedly, the main purpose of light reaction is having an input of water, and photons of light produce oxygen, ATP and NADPH. Of course, oxygen is being released as a byproduct, and in one of our previous lectures we really highlighted that this is one of the very big coincidences in nature, and a remarkable coincidence which is driving the entire aerobic life on this planet, just by the reaction of photolysis of water, that we are having oxygen being released in the process of photosynthesis, light reaction, only as a byproduct, having no direct role in photosynthesis process per se. So the light reaction, which is going to aim at producing oxygen, ATP and of course NADPH, that is the reduced coenzyme 2, would have to undergo the three very important steps of light reactions. The first step, of course, would mean the beginning or the photoexcitation. That is, the light from solar sources is going to be bombarded onto a plant in the form of packets of light quanta or the photons. This solar energy will be used to energize processes which are going to ultimately split water and release electrons. This whole process would be followed by the electron transport. Now the energy of the electrons will be harnessed to operate a proton pump, and this proton pump would work against the concentration gradient, and this is a type of an electrochemical gradient, and as a result the third part, that is the process of chemiosmosis, which is taking the advantage of the formation or the generation of a proton motive force and an electrochemical gradient, would ultimately result in the generation of ATP. Now the formation of ATP in the presence of sunlight is called photophosphorylation, a term which was given by D.I. Arnon, and who also gave the term reducing power, that is the formation of NADPH. Together, photophosphorylation and the reducing power, they constitute the assimilatory power. So here is a situation which is all or none. The next part of photosynthesis, that is the dark reaction, would ensue only if the entire assimilatory power is made. We will talk about cyclic and non-cyclic photophosphorylation when we delve into further details of the light reaction. Now, but first we have to begin with expanding upon a very loosely used term light bombardment. I used it myself a few minutes back. So when it is not sufficient to tell a student as to when light falls on a leaf. Now, how does it fall? How much does it fall? What is the quality of light? How much of it is absorbed? How much of it is reflected back? And are there other phenomena that it triggers? So that means one needs to know about the details of the nature of light, which is the basic topic of today. So everything in photosynthesis would begin with the pioneering process or the initial process of photoexcitation, which means the absorption of light photons, and somehow this excitation has to ultimately split water and release electrons. The other part of the story would continue only later. If we look at some of the very important statistics which are to be known and appreciated regarding the solar energy, it would be worthwhile to know that there are tremendous processes of fusion and transmutation going on the surface of the sun, and recently a NASA probe is going to touch the sun perhaps. So the amount of energy which is released, the entire energy cannot be focused towards Earth. If it was, then Earth would have been as burning as sun. So that is not to be. So it's only 40% of the total solar energy which is generated there is ultimately reaching our planet. And it has been calculated, it is around 5 into 10 to the power 20 kilocalories, which is enormous. If we like to define light in its physical terms, it is a particle, and it's also a form of radiant energy. That means there are so many forms of energy which are interconvertible, but this light energy itself is in the form of a energy packet, which we call as a photon. And what would be the nature of a photon? One needs to appreciate that it's not a photon which is carrying just one particular amount of energy. That means it would be the solar energy would comprise of a broad range of many such photons. Each of these photons would be associated with a characteristic amount of energy. So that means different energy possessing photons are falling on Earth. How do we categorize them? Its energy has to be categorized in terms of two important variables. That is the frequency V and the wavelength lambda, which is going to define its entire wave character. And this is how we would talk in terms of strong, weak, and lesser wavelengths, less energy, higher wavelengths, and so on. So the energy of quanta, when mathematically viewed, is according to the equation E is equal to hv, where h equals Planck's constant and V stands for the frequency of quanta, or we could even signify this whole equation of energy in terms of E is equal to hc by lambda, where again h is going to be Planck's constant and c would mean the speed of light, and of course the lambda the wavelength of light. So the entire energy spectrum which is coming from solar sources is going to be constituted of a wide variety of energy segments, and it would start from radio waves to gamma rays. If we see the spectrum on the left hand side, we have the radio waves which have a very, very high wavelength and extremely low frequency, followed by microwave, the infrared, and if you can see a very, very narrow visible spectrum or a visible range, which we would talk in details, followed by ultraviolet, X-rays and gamma rays. So that means these are going to be classified on the basis of their frequency or the wavelength range. In the depicted diagram, it is very clear that the gamma rays have minimum wavelength and therefore the highest frequency. Naturally, they should have the highest energy, and you know about the gamma cells and the properties of gamma rays as compared to radio waves. If we look at the electromagnetic spectrum in a little more details, we would find that in fact wavelength and frequency of a particular radiation are inversely related. So that means shorter the wavelength, higher would be the energy which is associated with it. And on that account, the gamma rays are the shortest in terms of their wavelength, and the radio waves, they are much longer. So normally a spectrum is made from by an increasing wavelength from left to the right hand side, and out of these the visible spectrum, as we would see a little later also, starts from 400 to 700 nanometers and depicting those 7 rainbow colors of the visible spectrum. So that means out of the entire range of the sunlight which is falling on sun, the visible spectrum that we can actually perceive with the human eye is just a very small band starting from 400 to 700 nanometers. So that means it is within this band of 400 to 700 nanometers that we can appreciate the different hues, and the world looks colorful to us, and the different light quanta would be associated with the different types of the color hues that we are having. Specifically to the visible spectrum, which starts as VIBGYOR, the violet part of the spectrum would be having the highest energy as far as the visible ray is concerned, and the red light, which is in the region of 650 until 700, would have the highest wavelength. It will be on the right hand side. Highest wavelength here would mean least frequency among the visible spectrum. This is the spectrum which is actually the range in which photosynthesis operates. We do not mention of photosynthesis taking place in an atmosphere of gamma rays or likewise in radio waves. So it is just this visible spectrum which is starting from 400 to 700 which is responsible for the photosynthesis, and it is called as photosynthetically active radiation, or abbreviated as PAR. So to summarize the entire idea which we have tried to crystallize so far, light is a form of energy with different wavelengths. The shorter the wavelength, the greater would be the energy which would be associated with the photon of light, and only certain wavelengths of light are detectable by human eyes, and they are appreciated by us as colors, and this would be the PAR, or photosynthetically active region of visible spectrum, and it is this particular visible light which drives the process of photosynthesis. This is the qualitative part. Talking about the quantitative part, we have to see that the entire 40% of the solar energy which was falling on sun, if the entire energy was being absorbed by a leaf, the vegetation and the planet would have been burnt many times over. But that is not to be. So that means out of the total light which is falling on leaves and the other green plants of a plot, just a very small portion would be actually utilized by the plant to trigger the process of photosynthesis. However, a major portion of the light is reflected back. Only a little portion is transmitted. So that means out of the entire gambit of the spectrum, electromagnetic spectrum, just a small PAR portion has minimized the amount of light which is actually being available for useful photosynthesis. And the reflection is so high, which is going to make us see the vegetation in different colors, usually green, because they reflect green, and some of it is transmitted, and the one which is absorbed has a useful fate in the process of photosynthesis. We would be discussing after a little break how the light which is actually being passing onto the chloroplasts or to the chlorophyll has a fate which is entirely spectacular, and just 1% of the total light which was falling on leaves, how is it utilized to perform the useful work of photosynthesis in the light reaction.
Thank you so much, sir, for this session. Dear friends, we will be continuing with the same topic in a short while, so stay tuned and keep watching. Thank you, sir.
Thank you.
Welcome friends, let's continue with the session what we are having in the first half of this lecture session, that was on mechanism of photosynthesis, and we were talking on nature and fate of light in the process of photosynthesis. So let's continue with Dr. Eklavya Chauhan with the topic. So sir, please continue the lecture.
Thank you so much. Viewers, you remember just before the break we had given ourselves a notion that the PAR or the photosynthetically active radiation is the only portion of an entire gambit of the electromagnetic spectrum where photosynthesis could exist. But do you remember the discovery which we discussed on one of the last lectures that I took, and the latest discovery was that is it possible to have photosynthesis beyond the red limit? You remember this discovery pertains to June this year itself, where certain cyanobacteria have been discovered, which is a remarkable discovery because these bacteria, they will shut down their chlorophyll A containing systems if the lights are say beyond 700 in the range of infrared, and another alternative form of chlorophyll, namely chlorophyll F, takes over. So this work was published in the journal Science by Professor William Rutherford and his colleagues, a very path-breaking discovery which prompts us now to think that photosynthesis is possible beyond this PAR. So we have to perhaps enlarge the range of PAR to include these exceptions also. To continue with the PAR itself, a biologist would always characterize different forms of light within the photosynthetically active radiation spectrum as wavelengths. So we find that in different wavelengths, the photons of different wavelengths would have different forms of energy. For example, blue light photons would have an energy value of 274 kilojoules per mole. Likewise, in sharp contrast now, the red light, which is on the right hand side of the spectrum, as I said, having less energy and a higher wavelength and a lesser frequency, of course, would possess just 181 kilojoules as compared to 274. And this is the reason why there exists a relationship between the number of moles of photons of a particular wavelength needed to excite a mole of a particular chlorophyll molecule. This is important because the blue light could excite a chlorophyll molecule much faster as compared to those corresponding red light photons, simply because of the fact that the former has a higher energy value. So in any case, the first step of any photobiological event has to be absorption of these photons by a photosensitive pigment. And the second vital question is, what is the amount or the degree of absorption that this particular photosensitive pigment or a chromophore is engaged in? How do we find it out? How do we quantify it? So this would be done with the help of a spectrophotometer, which can measure directly the degree of absorption. And when we plot a graph which depicts the absorption as a function of wavelength, we constitute an absorption spectrum. It is sufficient to say that the absorption spectrum of all different photosensitive pigments is going to be different and unique in themselves. So unique that it is something like a fingerprint that we have, because no two fingerprints of individuals can exactly match. Likewise, the absorption spectra of any two pigments can never exactly overlap. So this can be used as a very, very important key for the identification of pigments if one doesn't know what these pigments are. When we ask ourselves a question as to why do different pigments show different colors, you are familiar with the fact, and we also discussed it in our previous lectures, that chlorophyll A is olive green in color, the chlorophyll B is light green in color, and the carotenoids, they are yellowish, and chlorophyll F itself is in a reddish hue. Imagine a chlorophyll having a reddish tinge in its appearance. Now this is, the color is simply because what are the wavelengths which are reflecting back from that particular pigment, and that is being perceived by our eyes. So anything which is green means, it does not mean that it is absorbing green, it means that it is reflecting green. So the absorption spectrum of chlorophyll shows us that chlorophyll A has a peak in the blue region and a very strong one. You remember in the previous lectures we called it as a sonnet band. It was a very thick absorption here, and then there's a sleeve, and finally another peak comes around 660 nanometers. Likewise, we have the different absorption spectra for chlorophyll B and also likewise for the carotenoids. So that means the chlorophyll A is having two important peaks of absorption, both in blue light as well as in red light. The absorption spectra of carotenoids, of course, is quite different, because one would appreciate that if chlorophyll A is going to absorb somewhere in the blue region, the carotenoids are having a peak around 440 to around 510. So these are the regions which are in fact not operated upon by chlorophyll A at all. So that means these are the accessory pigments. They are going to help the chlorophylls by absorbing the light quanta in wavelengths which are not covered by the chlorophylls. In addition, if they appear to us in different hues of pink, orange and red, you should remember what happens in the deciduous trees, in the temperate zones in autumn, where we find that the chlorophyll pigments have broken down, the carotenoids are present, and they would allow the reflection of orange and red wavelengths, making the entire forest look in different shades of red as well as green. Now, as far as the chlorophylls are concerned, in photosynthesis, we find that the absorption peaks of different components are having a particular range. The chlorophyll A, which appears to us as blue-green, would mean that it is reflecting this color, and it operates at 430 and around 660 nanometers. Chlorophyll B is having nearly a similar absorption spectrum, but not identical, as I mentioned earlier. The phycocyanins are different. The phycoerythrins are themselves appearing as red. Now that's very important, because red is a wavelength which is not very strong in terms of energy, and the carotenoids are yellow to orange, and they also operate in different waves. This is particularly interesting to explain as to why there are different strata of plants which are growing underwater, because if you see carefully, the green algae are going to be present in very superficial waters, not deep beyond. The logic is very simple, because the green plants would have a preference for red coloration, and the green algae, they are not in a position to receive much of the red light if they are much deeper. The blue light penetrates much deeper as compared to the red, and therefore the red algae can be present much deeper in the oceans. They would absorb the blue radiations and they would reflect the light as red light. So that means the distribution of algae with different photosynthetic pigments has to be there, because we cannot expect the chlorophylls to be present in red algae in abundance. Because if only chlorophylls are there, what are the wavelengths they are going to absorb? They will not absorb the wavelengths which are needed or which are exactly available at such a high depth. So that means this is the phenomenon what we always call as a phenomenon of chromatic adaptation, or Gaidukov's phenomenon. In sharp contrast of the absorption spectrum, now let us look at what is action spectrum, because absorbing of light quanta by a pigment is one thing. To be able to act photosynthetically or operatively at a particular wavelength is something different. The question that we are asking ourselves is, are these pigments working at the same wavelength at which they are absorbing the light quanta? Perhaps yes, and perhaps not so much. So that means the relative activity of a physiological process of different parts would be how does it support photosynthesis. That means which wavelengths are the most effective to support the process, and we find that chlorophyll A is unique, because out of all the pigments, the chlorophyll A very well matches its action as well as its absorption spectra. So that means the wavelengths at which light is being absorbed are the wavelengths at which it is also most effective. So this nearly overlapping absorption and action spectra are going to minimize the wastage, increase the efficiency, and it is this reason by which we call chlorophyll A as the most efficient and therefore the universal chlorophyll. If we were to appreciate the matching of the absorption and the action spectra, you would find that if this is the absorption spectrum of chlorophyll A, the action spectrum is also very much the same. The magnitude is different, but then the wavelengths are nearly the same. And likewise, we have the chlorophyll, the second peak of chlorophyll A, and this is the action spectrum for chlorophyll A. So they quite match, and this type of matching may not be there for the other pigments to that magnitude. Therefore, it's a case of natural selection, simple natural selection, where nature has selected chlorophyll A to be the universal chlorophyll. If one looks at the different stages of absorption of light of the chlorophyll molecule, then we are surprised that how a particular molecule of chlorophyll is going to absorb the light quanta in an unbelievably quick succession, because needless to say, a molecule in a ground state would have the lowest energy. The moment it absorbs light, it would become activated, it would be photo excited, and imagine the process is such a rapid process that it takes just 1 femtosecond to be absorbed. A femtosecond is 10 to the power minus 15 seconds, whereas the photo excitation after 1 femtosecond is going to take the chlorophyll to a high energy state where other reactions will take place. According to the conventional Stark-Einstein law, it was believed, it is still believed, that only one photon can excite just one electron, and this is also called as the law of photochemical equivalence. But now the question that we have to ask ourselves is, are the processes of photoexcitation of a chlorophyll molecule strictly compliant with the Stark-Einstein law? Perhaps not, because chlorophyll itself is a very complex molecule and it has many electrons having different energy states. So that means each electron belonging to different types of chlorophylls has different capability of absorption of photons of different wavelengths. In other words, when electrons are exciting a chlorophyll molecule, that means there are different excitation levels to which these electrons have to go, and these different excitation levels are called as the singlet stages. these different levels would again depend upon what was the source of energization. If blue light was to induce excitation, things would be different. So if we look again at a mathematical expression, the energy of a photon is inversely proportional to the wavelength. So that means if a chlorophyll molecule is exposed to white light, which means sunlight, which has an entire gambit of spectrum, then we would find that there are different excited states at a given point of time itself. For example, blue light has a very high excitation of energy level, which is much greater than that of its red light counterpart. The blue light has about 58,000 calories per Einstein energy level as compared to just 43,000 calories per Einstein. And incidentally, 1 Einstein equals 6.03 into 10 to the power 23 photons. So it would mean that when blue light excites chlorophyll, then things are going to be different, higher energy levels are going to be achieved, and when red light is going to be the inducing factor, then things are going to be at a much lower ebb. For example, when blue light falls on chlorophyll, then it elevates, it excites the molecule to a higher energy level. That is, now these electrons, they would go to much higher energy levels, and this energy level is called as the second excited singlet stage. So the second excited singlet stage is a very unstable stage. It's an extremely short-lived stage. Its half-life has been calculated to be just 1 picosecond, and incidentally an picosecond is equivalent to 10 to the power minus 12 seconds. On the other hand, red light is inducing excitation in chlorophyll, but then to a slightly lower energy level, and that is, this lower energy level is called as the first excited singlet stage. However, this stage is also unstable, but not as unstable as the previous one. It is also short-lived, half-life being 10 to the power minus 9 seconds, which means a nanosecond. So it's a story of pico and nanoseconds. But then what is going to happen after these excited electrons achieve a second excited singlet stage or a first excited singlet stage? There can be many options. If the excited molecules do not react chemically at all with any other molecule, then they have to lose their extra energy, because they have to in any case come back to the ground level. Now the question is, how is this extra energy lost? In what form is it lost? We are all familiar with the first law of thermodynamics. So that means the energy could change its different forms. So a very obvious alternative would be that this extra energy can be lost or dissipated in the form of heat, having a thermal reaction, or else it could be lost in terms of light, where we see some fluorescence, or there's another phenomenon which is called as inductive resonance, where there's a transfer of energy, but then there is no radiation at all. So it is called as radiationless transfer. Or, which is more important to us in the entire discussion, certain specific type of photochemical reactions take place out of the downward passage of these electrons, these excited electrons. As a result, the photochemical changes would bring about now an electron transfer, ultimately going or channelizing themselves through the chlorophyll antenna molecules to the primary reaction center, which can initiate electron transfer, and finally, as we discussed earlier, to the whole processes of photophosphorylation and reducing power. But then let us have a discussion on all these four alternatives. So the energy, if it is lost as heat, one would appreciate that the chlorophyll molecule would decay, and the higher excited state, it would go to the first one, that is the lowest excited state, and heat is going to be lost. This is called as thermal dissipation or thermal deactivation. One should appreciate the fact that there is no further loss of heat from lowest excited state to the ground state. This is because heat can be lost only between second and the first singlet stages, because the transition is so very rapid. Imagine the transition being from 10 to the power minus 14 to 10 to the power minus 13 seconds is so very rapid that there is no scope for energy being dissipated as light. So it's only the form of heat that it would decay. However, from the first singlet state, excited state, to the ground level, we could have a loss of energy which would result in re-emission of photons as light. So the electron decay of this type is called as fluorescence. And as I said before, fluorescence is possible only between the first excited singlet state and the ground state. So to summarize the whole idea, the excited state of the highest type, that is the second one, would result in a heat loss, whereas the thermal dissipation as well as fluorescence would be the hallmarks of a transition from the lowest excited state to the ground level. So that means till the light exposure is continuing, fluorescence will also be shown because it is a re-emission. And the second very important part is that the light which is now being emitted is definitely going to be of a longer wavelength. That means the transition is always going to be a shorter to a longer wavelength. That means from a high energy to a lower energy. To give you an example, if there is a blue light irradiance, the fluorescence would be red. If you remember, in our previous lectures, we had talked about the Stokes shift. The Stokes shift was that there is always a difference in wavelengths between the absorption and the emission spectra of the same electronic transition, and therefore the fluorescence would always signify a higher wavelength. That means a lesser energy value, a lesser frequency. So this phenomenon has been named after George C. Stokes. The second option which is possible is the resonance, as I said earlier, the radiation-less transfer. So that means if there is a transition from the first excited state to the ground level through a special type of transfer which relies mostly on the proximity. So it's a phenomenon which is so very sensitive to distance. It is a radiation-less transfer between two light sensitive molecules or the chromophores. It is also called as FRET, that is the Forrester resonance energy transfer, and it is so very efficient that proximity is required, and the efficiency is inversely proportional to the sixth power of the distance between the donor and the acceptor. And if they are beyond a certain limit, then radiation transfer does not take place. Later, when we talk about the intricacies and details of the funneling effect of the chlorophyll molecule antennae and the primary reaction center, we would dwell at length as to how the photons now converge in the form of resonance transfer from the antenna molecules to the primary reaction center, which is P700 or P680 in the two photosystems. However, if all the three of the options are there, we would find that the excited molecule can return to another level or a stage which is called as the triplet or the metastable triplet state. Unlike the previous two stages, that is the second singlet and the first singlet, this particular metastable triplet stage is long lived, which is only 10 to the power minus 3 seconds. This is quite a high time period for different types of chemical reactions to take place, and the release of this energy can also be channelized in the form of re-emission, but then it would be called as phosphorescence. So the phosphorescence would be different from fluorescence because phosphorescence could continue even after excitation. So to summarize the entire story, we would find that the metastable triplet stage is the one which is giving sufficient time to the electrons to undergo chemical reactions, which we would call as the photochemical reactions, and they would ultimately result in photooxidation of the chlorophyll A molecule. So just to wind up the whole idea, we would say that the photochemical reaction is the fastest, and this is the best alternative and the most useful reaction to initiate the process of photosynthesis. Emission of light, emission of heat, emission of fluorescence, they are good in their own respects, but they are not useful to the process of photosynthesis. What one requires is photooxidation of water, release of electrons, reducing power to go to the next step of photosynthesis. So photosynthesis would start in real earnest when the chlorophyll molecule gets excited, and the photochemical reactions would harness the energy which is being dissipated in their journey from an excited state to the ground level. In the next lecture, we would proceed and take a lead from this very step. Thank you.
Thank you so much, sir. Your lecture is very interesting and knowledgeable. I am sure students are benefited from your lecture. And dear students, in the next lecture with the sir, we will be continuing with the same topic, mechanism of photosynthesis, part 2. So stay tuned and keep watching. Thank you so much. Thank you, sir.